Okay, so we are going to start with Chapter 1, an introduction to microbes. The and their building blocks part we are going to leave out. It'll come in the second unit with Chapter 6 and 7, and will actually be integrated in with Chapter 6 and 7. Okay. So what we're going to talk about is an introduction to the microorganisms themselves. Microbiology basically deals with all types of living organisms too small to be seen without magnification. In other words, you are going to have to use a microscope to see the vast majority of these. Now, there are a few exceptions to this, and we'll talk about why they're included in with the microorganisms a little bit later. Microorganisms will include bacteria, the archaeans, fungi, protozoa, helminths, and algae. Viruses will also be included in the study of microbiology, but they tread the line between being living and non-living. So we'll talk about that a little bit more when we get into the viruses chapter. That's chapter 5. Technically, I would throw them into the category of not being living organisms, but they do have an impact and are passed very similarly and have similar effects of the normal microorganisms that we talk about, things like bacteria. Right? So, as we're going through this, there are a couple of things that make studying microorganisms very easy as well as very difficult. The ease comes in when we talk about reproduction rates. We can get lots of bacteria very quickly. They reproduce rapidly. It's easy to get large numbers. What that means for us, and the bonus here, is that if you mess up, you can always go back and do it again. Okay. Chances are you can come back the next day and have an equally large population that you would have had the day before. The difficult part comes in the fact that you're going to have to use a microscope. You cannot see them directly. We cannot measure their life processes directly. Instead, we usually use what we term indirect measurement, meaning that we cannot see these organisms physically pick up and eat a starch or a carbohydrate, but we can actually measure the effects of what happens when they eat a carbohydrate or what happens when they digest certain types of carbohydrates. We'll see a lot of this in lab, so be prepared to see a lot of indirect measurements when it comes to lab. Okay. So what we're going to start off with is microorganisms and the planet. Now, I'm actually going to skip down to the second half of this slide and talk about that big bold print term that you see there. And let me switch over to my pointer really quickly. I like to have the pen. Okay. That big bold print word right there, ubiquitous. Okay. Learn how to say it. It is a very commonly used word in the scientists. It is a fancy term meaning everywhere. You can find them all over. In fact, there is a protein that is so common within your body, it is actually named ubiquitin. Okay? We find microorganisms everywhere on Earth in extremely large numbers, and they are very good at adapting to some of the strangest environments out there. We'll find them in places where other microorganisms cannot live. To be very honest, they outnumber you cell for cell in your own body. When you start looking at the number of cells that you are made up of, and the number is somewhere around 10 trillion, this has kind of changed as time has gone on. 10 trillion is the consensus, or has been up until pretty recently, and we sort of started upping the number slightly. And when you start looking at the number of microorganisms, there are about 100 trillion microorganisms living on or in you at all times. Now, what this means is that we are a decent home for these types of organisms. We're not sick all the time. They're not necessarily pathogens or what we consider disease-causing microorganisms. We're happy with them there, and that's completely fine. In fact, they actually have some pretty good benefits when it comes to our health that we'll get to in chapter 11. Okay? Now, the upper part of this, with the organisms being evolutionarily important, They fit nicely into the theory of evolution, mainly because we're pretty sure they were the first things available, the only types of organisms that were around for a very long time. And we're going to come back to this in a minute, what we are terming endosymbiotic theory. They've sort of been placed into a really excellent part of our evolutionary history. So let's talk a little bit. We're gonna go way back, billions of years back, and we're gonna talk a little bit about sort of evolution and microorganisms. Okay. So we are quite sure that there were bacterial type organisms, basically very, very simplified single-celled organisms that were around about somewhere between 4 and 3.5 billion years ago, and they were the only things around for around 2 billion years. Okay. These organisms we describe with this term. We call them prokaryotes. Okay. If you don't know, I like words. So let's talk a little bit about what it means to be a prokaryote. Well, pro means before, and this word karyote comes from the word karyon. It means nut. Okay. Now, believe it or not, this is slang. Okay. Because the first people who looked through microscopes and were seeing individual cells through the microscopes actually realized that some of them have little walnut-like structures on the inside while others did not. I always think of these as the mounds and almond joys of cells, because sometimes you feel like a nut, sometimes you don't. Yes, I make cheesy jokes, and yes, you are expected to laugh at home. So the prokaryotes before the nut, they have no true nucleus. The reality is it's not just about the nucleus, but the nucleus is sort of the big defining characteristic. Prokaryotes have no membrane-bound organelles, and the nucleus is included in that. Your nucleus is a membrane-bound structure. Excuse me. Right. The other cell type we see, and still to this day, the other type of cells that are out there are what we term eukaryotes, and that term "eu" means true. Okay? These are organisms that have a true nucleus. They actually have that nice defined membrane-bound nuclear structure on the inside. Okay? So these we consider precursors to the organisms that eventually became multicellular. The eukaryotes began living together. They lived colonially. When you put cells together, they form tissues, and tissues together form organs, and organs form organ systems that are made to form organisms. So these were our precursors to large multicellular organisms. Now, the sort of how is the next question, and that's what always gets me. Somebody always says, well, how do you know that? How did that happen? Well, we're going to back up a slide. Okay. All right. And part of what we think actually happened is that, and I'm going to throw screen in here. Okay. And part of what we actually think happened is that in the very beginning, everything was considered a prokaryote. There were no membrane-bound organelles in any of the types of cells that were out there. Okay? Now, part of this also means that there were a lot of different types of prokaryotes out there. Okay? Some large, some small. Okay? And one thing we know, and it's still true today, is that the larger you are, the more energy needs you are going to have. Now, because the larger prokaryotes were going to need more energy, a very, very good source of energy would actually be those smaller prokaryotes. So, the larger prokaryotes actually started ingesting the smaller prokaryotes to use as a food source. Now, part of the bonus to this actually came in with the realization that these smaller prokaryotes, once inside of the larger prokaryotes, I often say that they got Pac-Man. I like to do this. Okay, so that the large prokaryote came along and grabbed hold of the small prokaryote, but instead of digesting it for its energy, digesting it for things like carbohydrates and carbon sources and energy source, instead of digesting these small prokaryotes said, hey, wait a minute, okay, just give us a chance. If we sit in here long enough, this large prokaryote suddenly realized that those small prokaryotes sitting inside were actually making life much easier by helping to produce large amounts of ATP. If you remember your biology, ATP is code for usable cellular energy. Okay? And instead of actually digesting these, okay, these cells had a lot more ATP, okay, and were a good bit more successful. So these smaller prokaryotes actually ended up becoming organelles inside of the larger prokaryotes. Okay. So we start talking about evidence for this. What we see is that within mitochondria and chloroplasts, evidence for endosymbiotic theory, and I definitely want you to remember this one is a great testable term. Evidence for endosymbiotic theory is that mitochondria and chloroplasts actually look a lot like bacteria. In fact, within eukaryotic cells, mitochondria and chloroplasts actually have their own DNA, and it is structured very similar. It's actually structured exactly like bacterial DNA. They have their own ribosomes, again, structured pretty much exactly like bacterial ribosomes. And if you remember, the bacteria are prokaryotes. And they can replicate independently of the cell. The cell itself does not have to force replication for mitochondria and chloroplasts. They can replicate all on their own. Okay? So there's a little evidence of endosymbiosis or endosymbiotic theory. These basically prokaryotes that are living as organelles inside of eukaryotes. Aside from being major players in the role of the evolution towards eukaryotes, we also see microorganisms shape the planet greatly. Based upon all of our evidence, we know that the first conversions of energy were probably photosynthetic conversions. We were probably still doing photosynthesis and capturing light and repurposing it as a form of energy. But it was probably what we consider anoxygenic photosynthesis. In other words, photosynthesis that does not produce oxygen. Okay? Eventually, there was a shift to oxygenic photosynthesis, and photosynthesis actually began producing oxygen. This was a huge bonus to the eukaryotes and other even unicellular organisms, because the oxygen in the atmosphere actually allowed us to make more energy. The more energy organisms have, the more things that they can do, the more reactions they can run, the more movement they can do. So oxygen photosynthesis was a huge, huge bonus when it came to the energy needs and energy demands of organisms on Earth. Still to this day, photosynthetic microorganisms account for around 70% of the Earth's photosynthesis. So about 70% of our oxygen that is a product of photosynthesis comes from microorganisms. It's mainly things like algae in the oceans. That's why we very often caution people against polluting the oceans, because when those algae begin to die, that's when we're going to see a lot of oxygen depletion. Okay? Microorganisms also drive the structure of the soil and the water and the atmosphere itself. Aside from oxygen, they produce other very important environmental gases. We see lots of fungi, lots of bacteria actually living in very close associations with plants, being part of the nitrogen cycle, helping to recycle nutrients back into the soil, back to the earth itself. They're very, very good at helping us maintain the environment that we have. Okay. Now, I'm not only going to stick to sort of environmental microorganisms. We are going to talk about microorganisms in humans. Historically, we've seen microorganisms used for production of bread, alcohols, cheese. Those are three of the best things on earth. You give me bread and cheese and I'm there. Mainly these are a product of fungi. We do see some bacterial use when it comes to the cheeses. It depends upon the cheese. Treatment of wounds and lesions. Antibiotics are actually products of other types of microorganisms that are used to ward off one type of microorganism. So we get it from one organism and it's used to ward off a different type. Mining precious metals. Gold is actually mined with the help of cyanide, I believe it is, and the toxins that are used to displace the gold from the rock that is in the gold from the ore have to be gotten rid of. They use microorganisms to do that. This is a process known as bioremediation. It actually shows up on the next slide. And this includes the cleaning of human-created contaminants, things like oil spills or wastewater. Genetic engineering and the recombinant DNA technology, very often, honestly, these are combined. Okay? One is not necessarily used without the other. When we talk about genetic engineering, we're talking about manipulating the genes of an organism. This is commonly done via recombinant DNA technology, so these go hand in hand, where we transfer genetic material from one organism to another in an attempt to sort of deliberately use that DNA. Probably one of the best examples of this is insulin. Okay? If you know an insulin-dependent diabetic, one thing that is very interesting is that insulin used to not be particularly easy to get, and it was extremely expensive because insulin was extracted from pigs. It was porcine insulin. Now, when you buy a vial of insulin, it is actually human insulin. The insulin itself is grown in bacteria. We're not extracting insulin from other humans. Instead, what we've done is take a piece of DNA that codes for how to make insulin, okay, and make copies of it. So we've taken the gene for here's how to make human insulin, made copies of it, and inserted it into the bacteria. Bacteria are actually very good at uptaking and utilizing foreign DNA. So the bacteria pull in the DNA, begin to actually use it to make insulin. Okay? And what we've done is sort of tricked the bacteria into producing human insulin. Okay? Now, this is much cheaper. Again, you can grow lots of bacteria very rapidly. It seems to be an extremely good way to deal with the problem of things like the expensive insulin and people that have allergies to the porcine insulin and all the processing and the waste that was occurring. We have made insulin much, much more accessible, usable, and cheaper. Okay? Again, the bioremediation, like we were talking about, and the use of microorganisms to clean up contaminants, things like oil spills, or wastewater contamination or runoff contamination, because there are microorganisms out there that digest unusual substances or live in unusual substances. What that means is that those organisms are going to have to be able to break those substances down. Because those organisms can break those substances down, they're really, really useful when it comes to removing those substances from an area. And the nice part about it is they're kind of self-cleaning. It's sort of the scrubbing bubbles mentality. If we pick up a bacteria that digests cyanide and put it in a cyanide pool, once that bacteria has metabolized all of that cyanide, it's a self-limiting cleanup because the cyanide is gone. The bacteria will then also die because they've pretty much eliminated the food source that they were growing on, unless we keep adding back more cyanide and keep using those organisms to get rid of those particular contaminants. Now, aside from all of the good that I have talked about, we cannot get around the fact that microorganisms do harm humans. We are going to talk and spend a lot of time talking about pathogens. Okay? But be aware, the majority of microorganisms associated with humans are non-pathogenic. Remember those 100 trillion that were living on or in us at all times? For the most part, they're helpful and not harmful. But the pathogens that are out there, and pathogens is a term for microorganisms that cause disease. The pathogens that are out there are responsible for a lot of infections. Okay? And they can be extremely dangerous. You do not have to memorize these numbers, but for your information, the World Health Organization estimates there are 10 billion new infections caused every year by microbes. Infectious diseases are among the most common causes of death in the United States and worldwide, with the death toll being approximately 13 million people per year worldwide. To put that in perspective, the CDC reports that a child dies from malaria every 30 seconds. That is a lot of infection, and it is extremely believable. To be honest, I would say that 10 billion new infections every year is probably lowballing it. Think about it. There are around 7 billion people on the earth. Each of us gets sick 1.5 times a year. That includes a cold. Okay? I definitely believe it. For those of you that aren't getting sick, those of us with children are making it up for you. Trust me. Okay? We also see a lot of emerging and re-emerging infectious diseases. The emerging diseases, those that are considered new, in the grand scheme of things, AIDS is a relatively new disease. Now, as far as us characterizing it and calling it AIDS, that happened in the early 80s. The reality is we have serum samples from the 50s that actually contain the HIV virus. Hepatitis C, you've seen these in your lifetime. SARS would be considered an emerging disease. Last year, Middle Eastern respiratory coronavirus, MERS-CoV. We're also seeing what we consider re-emerging diseases, the ones that we thought we had a handle on suddenly coming back. Measles outbreak in California, tuberculosis. We thought we had TB under control. It's not really the case. Now we're seeing more virulent, more dangerous pathogens that seem to have a lot more ability to resist the drugs that we're currently using. Okay? One other thing that is very interesting is that diseases that we used to consider non-infectious, in other words, they didn't have a link to a microorganism, have now actually been linked to microorganisms. Probably the best example, one of the first of these in the past 20, 30 years, are gastric ulcers. Helicobacter pylori is the bacteria responsible for causing stomach ulcers. And for a very long time, we thought it had a lot to do with genetics or what people ate. And in the reality, it's an infection. Okay? We see higher instances of diabetes in individuals that have had repeat infections with Coxsackie virus. Schizophrenia and Borna agent, which is another viral agent. About 75% of schizophrenics or their mothers actually test positive for antigens that prove they've been in contact with Borna agent. Now, why the link is there, we're still not quite sure, but there does seem to be some type of link. Okay. We see chronic infections with bacteria or viruses being linked to things like multiple sclerosis, heart disease, obesity, obsessive compulsive disorder. The more we know and the more we're studying, the more we seem to see links between things we've considered non-infectious and microbial organisms. Okay. So I am going to stop here for the first half of chapter 1. We're going to do a little bit of history and then go through a few of the cell types again in the second half of the chapter. The history is going to be brief, but I do want to divide this up just so you have sort of a nice stopping point.